Quorum sensing is a type of communication in bacteria that produces various sensing molecules that allow bacteria to communicate about population size, metabolic states, or produce an end product such as bioluminescence. Quorum sensing is apparent in both single species populations and mixed species populations. It can be used as a decision-making tool in which individuals have the capacity to assess the number of other components they interact with and have a standard response once a threshold number of individuals is reached. Acyl homoserine lactones are present mainly in gram-negative bacteria and are in control of their own synthesis. There is a lot of diversity in this group, with different bacteria species being able to produce unique lactones. However, some bacteria species produce the same lactones. The head group consists of the homoserine lactone and is conserved among all types of lactones. The tail group is the acyl function group and is a variable region that determines the specificity of the receptor. Oligopeptide molecules are present mainly in gram-positive bacteria. Their synthesis is dependent on ribosomes and their peptides can be modified. Autoinducer 2 is a quorum sensing molecule used mainly by gram-negative bacteria to facilitate communication between different species. There are 3 mechanisms in which bacteria conduct quorum sensing. Acyl homoserine lactoses are synthesized by enzymes and move through the bacterium membrane via passive diffusion. When a significant concentration of lactoses is reached, it binds and activates a regulatory protein on the cell membrane. The regulatory protein then binds to a specific DNA site and influences the target gene. It also induces more production of the lactose signaling molecules. Oligopeptides are cleaved into small functional units and secreted out of the bacterium through a transporter protein. Signal oligopeptides are detected by a sensor protein on the bacterium's surface when they reach a significant concentration. The protein becomes phosphorylated and the phosphate is transferred to a response regulator protein. The phosphorylated regulator protein binds to a specific DNA site and alters transcription in the target gene. Autoinducer 2, also known as AI2, is formed by recycling the molecule S-adenosylmethionine in the adenyl methyl cycle until DPD is produced. DPD is then converted into one of 2 forms via hydration. AI2 passively diffuses through the membrane, and the LSR transporter brings AI2 into the cell. AI2 is then phosphorylated by LSRK. The AI2 phosphate binds to LSRR and causes it to release DNA, which decreases transcription of the LSR gene.